Hello and welcome. My name is Mitch Sanders. I'm a part of the Communications Committee along with Jordan Yarin. We're delighted to have another Wound Healing Society awardee. This is a travel award, and today we have a very special guest. He's Anish Vasan he's a Boston University PhD student. Anish, welcome. Jordan, thank you kindly for doing this kind work with us again. And we're delighted to have you and host you. And I'll let Jordan start out with the questions, and we'll go from there.
Thanks, Mitch. So Anish, the title of your abstract that you won the Travel Award for was a medium throughput 3D wound on a chip screening platform to assess provisional matrix assembly during stromal wound closure. Could you give us a brief overview of the work and why we should all be excited about it?
Sure. Well, I know that the title is a bit of a mouthful, but I think it accurately describes.
PhD scientists, it's not at all. It's very, very simple. It sounds wonderful, though.
It's a work in progress. But the unique thing that I like to point out about our system is that, well, it is a 3D wound on a chip, and it holds stromal tissue under tension, something that usually in vitro models lack the mechanical context of skin actually being under tension. And the additional thing in that it forces these stromal cells, the fibroblasts, to create new matrix before it can heal the wound, something that actually happens in our body. So compared to the classic scratch wound assay, where when you make the injury, your cells are migrating over a 2D surface that's rigid. This model actually allows you to study the process with which these cells create new matrix before migrating into the wound. And the idea we had with this was that if we could make it a high throughput system, then it would have a lot of applications in the industry in terms of like a drug screening platform, or just a methodology to study what goes on in the body with just a little more, shall I say, volume.
Wonderful.
And yeah, I called it medium throughput because in my mind, a high throughput system would need to be way more. But then after a recent conference, someone told me that it's already pretty high throughput, so I might change that in the future.
So how many cells do you need to do the assay? Is this something that you could isolate RNA from as well?
Well, our tissues are pretty small. Now, these are micro tissues. And so each tissue actually starts off with about 7,500 cells. So I would say like doing an RNA extraction from that is possible, but it's still a very small amount. You have to be very careful. You might have to pool samples instead. So there is that part of this, the assay that you might not be able to do. But what this does give you is a physiological readout and a physical readout, because you can actually stain or label the ECM in these tissues, and that gives you the spatial context and the temporal context, as opposed to a bulk context.
So let's talk about you before you started on this PhD. Sure. And where did you do your undergrad? How did you get here, and how did you get to be you?
Well, I did my undergrad in India at an institute called the Birla Institute of Technology and Science. We call it BITS. And it's in Rajasthan, which is a desert in India. So I'm actually pretty used to, like a very wide range of temperatures, going up to like 120 in the summer and sub 30 in the winter. So we actually did a double major in mechanical engineering and biology for my undergrad, and we were required to do like a 6-month thesis project, which was like a capstone project. And I had the opportunity to do that here in Boston at the Brigham and Women's Hospital in Dr. Hadi Shafi's lab, which is how I got into the whole diagnostics and microfluidics. I mean, that was my training, MEMS and biology, and I had the opportunity to apply it there.
So that was your Zika virus work, or what was that?
That was my Zika virus work. That was what my thesis was on. And I got hired by Dr. Shafi to work for a year after that as a research tech. And I saw that as a good opportunity to not only finish up my paper on the Zika virus diagnostic tool, but also to expand the work. And so I was able to come back and then continue the work for like HIV, HCV, CMV, and HPV. So like I was able to like expand that technology. And yeah, it was quite the experience.
So after several years in Boston, you still don't mind the cold, snowy weather? We're gonna have a bad winter this year. You may change your mind.
I love it. I love it. I mean, like I said, I'm used to a wide variety of, or rather, I should say, a wide range of temperatures, and I just learned, you can't really do anything about the weather. You can dress appropriately for it, and beyond that. But I had never seen snow before, and I guess I just fell in love with that. So I don't actually mind the winter as long as I have a good coat on.
So Jordan's university is beautiful, and they have the most amazing scientists, but the weather's hot.
And I guess that feels like hope.
I didn't see snow until my 20s either. So.
And I did my undergrad at Boston University.
Oh,
and my
lovely.
PhD at WPI, and then 2 postdocs at MIT. So I live down the street, and I work in Framingham. So you have to come to. We're right next door to Jack's Abbey, and you have to come over for lunch and give a talk. It'd be really fun. So we'll take you for beer and really good food.
Thank you for the invitation.
So tell us more about, is there a paper in the works? Is it already public? Is it pending publication, or where are you with the data?
If you're talking about the wound healing system, yes, we're working on it. I'm looking to expand it a little more in terms of just showcasing the capabilities of the system and just putting a little more rigor into it. My whole shtick with it is that I want to show that it can be used for mechanistic studies, which would be important apart from just the quick physiological readout, because while it is a screening system, it is also a good scientific tool, in my opinion. And so the paper I'm trying to put together right now highlights both aspects of that.
And just so you know, I'm doing, and now I feel, I don't want to say incompetent, but I'm doing scratch assays, and I'd rather use your system instead of that.
Oh,
but
wow.
I have a new class of drugs that we're actually using for a whole number of indications. And we figured out what we think is the kind of business end of the molecule. And so we found peptides that we think will work, as well as drugs that may mimic the structural benefit of the peptide and the actual protein. So we'd love to figure out how we could collaborate on, under an NDA, figuring out whether this could work for our system as well. And with this is a program that we're doing, and Jordan knows, we're doing everything from traumatic brain injury to wound healing with this class of molecules. But with the last couple of months, since I gave a talk at Jordan's at ASU, we've already figured out the business end of the structural domains that are involved. And we've already started designing just peptides, as well as we've found a class of drugs that are already on the market that may actually have potential opportunities to do the same thing. So other indications, which is really, really fun, because then the pathway to market is much faster. But tell me more about, so in which lab did you do the work in, and what are your goals? What are your future goals?
So this work was done in Dr. Christopher Chen and Jeroen Ikman's labs. I am co-advised by both of them, and so I share their resources. And so they're both at BU, and yeah, I mean, this is based on work that Chris and Jeroen have done in the past. In fact, this model system has been published about before. The novel addition being a slight change in geometry that makes these tissues viable longer, and also the high throughput model, something that, like, at least earlier we did not have. So as for my future, well, I am thinking of academia. I started the PhD program being 100% sure. I'm now evaluating, whether or not that is the route for me. But I'm still, leaning towards it. but I'm sure like you probably know this just as well in terms of like the doubts that you face when you're doing your PhD. And I'm just working through that right now. So it's lovely to see, and always talk to people who are enthusiastic about research in the academic space and who are especially interested in translation, because that is something that I am quite enthusiastic about.
So Jordan is probably one of the smartest guys I know, and going to ASU could be awesome for you. But I want to talk about the dark side. I work in industry, and I love what I do, but I also get to develop cool things and write patents and write publications. So it's not just, if you go to industry, it's not boring, certainly not at all. And but I would pursue both. When I finished my PhD, I thought I was going to take a job at University of Hawaii, but I didn't get the job, and then I took a job in industry, and off I went. But I haven't. Just talking about Jordan, he's a superb scientist, and ASU is one of the best programs I've ever seen, and the people they have on staff is just unreal. So I'd like you to apply to both, but I won't feel bad if Jordan offers you a job before I do.
Well, I can't say I have decided, so I will definitely apply to both.
Very cool. And can you talk a little bit more about the microfluidics work and the diagnostic work that you did on Zika as well?
Sure. Yeah. We published that paper, I think it was last year. So it was a nanoparticle-based system. Well, this is one of the works that we published that is the most close to my heart, because that was the one that I started and saw through till the end. And essentially, we built a cell phone microscope, and we built these microfluidic chips that you could essentially just put a drop of blood in, and it would, basically have, it was like a lab on a chip that would then react to your, like, these blood samples with nanoparticles that had platinum in them, and with a reaction with H2O2, you'd form bubbles if there were, if there was a virus that it had captured in your sample. And so then you could read off quantitatively whether or not you had said virus. Now, the platinum nanoparticles were conjugated with the antibody specific to the spike protein for each of these viruses.
Sure.
And so my work focused on like the Zika virus, because that's when I came to the US. We started, targeting that system first, and then we expanded it on once we saw that, well, one, we weren't able to publish in time during the window that, everyone perceived as, yes, the most, what do you say, prime to publish about Zika. And so we realized that, okay, then, this system was capable of multiplexing. And so I built the system to have like multiple channels, and then you could just like put a single drop of blood, split it up between channels, and you could have a readout based on, okay, whether you have HIV, HCV, Zika, and so on. And yeah, I mean, I don't believe I've done a too good a job of explaining how the microfluidic works because it's been a while. But essentially, we use laser cutting and laminates, so like double-sided adhesive and acrylics to make these channels. So we didn't actually have to use microfab. And that also made this system really cheap because plastic. And you could cut it using a cutting plotter, or you could use a laser cutter. We used the laser cutter to do this. And the idea was that you could just use a simple solution of silane, so like Rain-X, for example, to make your plastic surfaces hydrophobic. And so then once you loaded this lab on a chip with all your reagents, it would just slide along on your channel, mix in the mixing chamber, and the reaction would go through. And I think one of the most challenging parts of that work was actually formulating a freeze drying method that we could preserve reactive chambers so that you could freeze dry them and then store them in these vacuum food saver bags for up to like 4 months, 5 months. That's as long as I tested, because that was one of the requirements one of the requests from the reviewers for our paper, to show that it was viable in terms of like long-term storage, because we had claimed it. And so it was very interesting to be able to like formulate like freeze drying methods and then store it, and then have to like take these bags out and like hope that it worked, because in the end, like when we say that, yes, theoretically, when you look at the science, it's sound. But when you're actually testing it out, there's always a question: did you mess up, the storage system?
Yeah,
Um,
one
but
of
yeah.
the first diagnostics I developed, it worked perfectly even for 6 months to a year until we went on a plane, and then we were seeing that we didn't realize that if it could get any moisture in, it was going to fail. And eventually we were able to get a 2-year shelf life. But on a little protease or a peptide-based diagnostic measuring a protease, you can imagine that water sensitivity was a huge. As soon as you got water in the system, it would eventually hydrolyze, and then your shelf life was toast. And it took us, we didn't realize until we flew to Sweden with the product. It said, oh, and we were in Gothenburg, and I don't know if you've ever been to northwest, it's southwestern part of Sweden. It rains every single day except for like 1 week out of the year.
Right.
And that realized that moisture was killing our diagnostic. It was really
Wow.
Yeah, I mean, it's interesting you brought that up, because that was actually one of the questions that we had in the review, because this system is quite viable in low resource settings, because once you have the chip, you don't really need much except for hydrogen peroxide,
Sure.
and say PBS. So you could even use water actually. And once you do the washes, you don't even need like the lab on a chip that I designed. You can actually just pipette stuff in, or just like force it in with a dropper into like the reaction chamber. And one of the questions we had was, okay, does it work at like different temperatures? Does it work at different moisture levels? And so that was actually something that I had to test, where I exposed the chips to different temperatures. And so I was like literally working in like a cold room. I worked in like one of our room size incubator rooms also to show that the chip was viable at different temperatures and different moistures as well. And I think because of the way the chemistry, like it was essentially simple click chemistry, but the antibodies were protected because of that chemistry on the surface. And so they were stable for quite some time. And so even if they were reconstituted with moisture in the air, it was still viable.
It's really cool. Jordan, I asked too much. What do you think? What do you want to ask?
No, I'm enjoying all of this. I started my research career doing rapid prototyping of microfluidics with CO2 lasers and adhesives and all that. So it's a walk down memory lane. I did want to ask a couple of questions back on your abstract. The device is very interesting, and we've in our group have become very interested in the biomechanics of skin and its relation to wound healing, and of course there's a lot of attention right now about mechanical offloading and its relation to scarless healing and things like that. So I'm curious, what level of control do you have on the mechanical strain on the samples that are grown on these chips? Is that a variable that you can tune so that you can mimic specific strains associated with healthy skin, or something like Ehlers-Danlos syndrome or something like that? And secondary to that, can you actually measure how much strain is on your tissues?
Right. So if I understood that right, two questions. One, can you control the strain? Two, can you measure the strain?
Right.
Right. So the control the strain part of it, I would say this specific system does not have that level of control, because you are limited in the 96 well plate system. So the application of strain, there are two ways to do it based on previously published work. One was done in our lab, where they placed magnetic microbeads on the pillars and then used magnetic tweezers to pry the pillars apart, and then you could control. So yes, actually that system might be applicable in this 96 well system. I just personally have never worked with it. This was work that was done before I joined the lab and was done in another form factor of these devices. The other way to do it is actually using air and vacuum as a way to deform the pillars from the base. And that is actually something, I'm not sure if they've published it yet. I think they have, by Alison Scrow, who is a collaborator who is here at BU. She just moved to Genelia. And so her system is capable of, like, they call it air tugs. Our system is called the microtugs, which was the original one. And so the air tugs essentially work on the principle that you can apply either air, like pressurized air, into these small chambers that are near the pillars that would force the pillars to deflect. So that one, then you could apply specific strain to the system. The alternative, of course, is like you can apply clamps around this and stretch. That is the most basic system, but then again, it's not viable for high throughput, nor is it exactly viable for bidirectional strain. But it's an interesting question that I've toyed with, but that's not currently the direction my research has taken. So
I see.
Yeah.
So you mentioned that these are
I'll just go quickly. The measurement part of it, you can actually measure the strain or rather the stress that the tissue is exerting. In that you can measure the pillar deflections, and from that you can back calculate the amount of strain and stress that the tissue is applying on the system. And this is again something that Wes Legand published earlier in the original microtug paper, because we know the material properties and the structure of these pillars, and so with that you can calculate forces.
Really?
Sorry, I spoke over you, but I just wanted to get that out.
Yeah, no, I wanted to. You were going on an interesting direction that we could talk for hours about this. I think it's a very interesting study. I really wanted to know about that measurement, because when you're in an optical system, of course, right? You're not in a 96-well plate. You don't really have the opportunity to stick in a lot of probes to start measuring things unless you develop a whole biomem system to do it. So maybe in the next iteration, you'll incorporate some things like that. It's very interesting. Do you have any plans for using this for screening at the moment? Do you have molecules you guys are working on to use the system?
Yes, I mean, in the capacity of like the biomechanics, just to explore like the involvement of the various forces and how they're exerted through these cells and the ECM. My primary focus is on the ECM, as the title also alludes to, the study of the provisional matrix generation. And so I'm interested in how like these cells exert forces on these ECMs. And so, like how do they pull it into the wound when they create it? How does it get aligned? All of that. So a lot of cell ECM interaction molecules. Specifically, as a screening platform, I do have an idea in terms of like the drugs that the class of drugs, but I haven't gotten that far yet, because my other part of the research also focuses on just, what I say, like the different cell types we could use in this system, and that's part of the presentation that I made, in that we could look at like adult dermal tissue, neonatal dermal tissue, cardiac fibroblast tissue, lung fibroblast tissues. And so like look at the differences in how those cell types create matrix. And so I've been focusing a lot of my work recently on that part of it, as opposed to the screening system.
How long does it take to generate a data? Is it a week, or how long does it take to?
From start to finish, let's see. It takes a day to make the plates. It takes it takes half an hour to seed a plate, and then it takes a day for the cells to self-assemble, because as Chris quite eloquently put it, we make the jungle gyms for the cells to play in, but they do their thing usually, and so we just guide them in that way. So the cells self-assemble into this microtissue. Next day you injure. Depending on your cell type, it takes a different amount of time for it to heal. And so that is again something that I was able to showcase, in that different cell types heal quite differently. Like the neonatals heal in about 24 to 26 hours. The adult dermal tissues actually take more than 72 hours to heal. And you can see that also in terms of like the difference in the ECM that they create, the amount of ECM that they create. So I just presented last week as a poster at BMES about this kind of quantification using a fluorescence system. I mean, ideally, I'd also like to use something like Western blot and qPCR, but it's still a work in progress.
I'm a big fan of nanostring, because then we can look. I've just made a new kind of wound panel that also includes fibrosis and a bunch of other things, and even macrophage plasticity. So I've got about 300 genes that I like to look at a time. And the cool with nanostring is you could also do spatial. You could look at the expression in situ in the tissue and compare the different cell types or compare the different regions of the leading edge versus the kind of the back part of the wound that's healing or the scar that's healing or the what have you. We'd love to find a way to fund a project with that for anywhere from a week to a month to better understand the molecules that we're testing. And it may be a more advanced version of the scratch assay that we don't love, and we'd love to get more information. So please reach out to us afterwards, and I'm sure Jordan
definitely
for a postdoc position by tomorrow.
Well, the postdoc position would have to wait. I am nowhere near graduating right now. I think it's going to take at least a year and a half, if I'm being optimistic.
We're not going there. We'll both be around and still hiring people by next year. So don't worry. Not a concern. But
collaboration-wise,
if you get a chance to go to ASU, I love the campus and the people there. And you can come down and just drink beer with us and see my lab in Framingham, whenever you want. Well, I know we've almost been on for an hour. I think Jordan's right. We could probably talk for several hours, but I think this was just a wonderful opportunity to congratulate you on your award. Thank you. We look forward to collaborating and potential opportunities in the future. And if you're, talk to your advisors. If you ever want me to come to my old stomping ground at BU and talk about all the wound care stuff I do, I'd be just honored and delighted.
Yeah, I would love that, and I will bring that up. If either of you is interested, we do have department talks from scientists, and I would love to have both of you here if you have the time. So I'll bring that up.
Let me know if you ever want to go to a Bruins or a Celtics game, because we can get tickets through the company and go goof off and enjoy a game. So
sounds good. And I will be reaching out about the nanostring stuff because I'm very interested.
Come up for a game. It'll be really fun. I promise.
Will do.
All right. Take care, Anish. Real pleasure meeting you. Jordan, thank you again. And look forward to having this hosted on the Wound Healing Society website very shortly.
Okay.
Take care. Cheers. Thank you.
Cheers. Okay. Bye. Thank you.
Bye.